Hello, and we now begin Module 9, and this is going to be about microbial ecology and diversity. The first part of the semester, which is everything we've done before, Modules 1 through 8, have probably kind of laid the groundwork to really starting to now investigate the impact of microorganisms in the world. In this section, we're going to talk about their impact in nature. In Module 10, we're going to start talking about their profound impact in host microbe interactions. Then in Modules 11, we're going to talk about how we defend ourselves against microorganisms. In Modules 12, we'll then talk about pathogenic microbes. And then finally in Module 13, we're going to talk about public health, how just government organizations try to manage and protect us from various public health threats, which right now, because of the coronavirus pandemic, is quite timely. Let's begin. Here's the module outline. We're first going to talk about, in this module, community physiology in any environment. We're then going to talk about the techniques that are used in microbial ecology. We'll then look at examples of microbial environments just to give you an idea of the patterns that you find. We're going to finish talking about cycling in elements, and we're going to talk about probably the two most important elements, nitrogen and carbon, but there's cycles for all the different elements in the environment. These are just the two that we have time to cover. Here are the learning outcomes. Again, those are basically for design documents. First of all, let's talk about what ecology is. Ecology is the study of how organisms interact with their environment. Microbes influence the ecosystem. They have both a metabolic activity, and this metabolic activity of microbes actually determines the Earth's biogeochemistry. In other words, what I'm trying to emphasize here is that microbial activity has macroscopic consequences. And you'll see examples of this as we go through. Ecology is concerned with the distribution and abundance of organisms in nature. In most ecology you're probably familiar with is looking at animals and plants in the environment. Microbial location is also important, and it is determined by how well they can grow. What's their metabolic potential? Are there things that they can use to generate energy, electrons in reducing power, and to make cell carbon? Also, what are the environmental conditions? Can they survive them? And then what other organisms are there? Can they build communities with them? Here is a really simplified diagram of the food webs with major roles for microbes. In the oceans, you have phytoplankton, bacteria, and algae, and they produce a lot of the biomass. They are the primary producers there, phytoplankton, bacteria, and algae. 60% of it is taken up and used by grazers, and these will mostly be protists and invertebrates. 40% is destroyed by viruses, and viruses are very important in recycling elements and keeping the amount of bacteria and algae under control. In the forest, the main primary producers of biomass are plants, trees, other bushes, etc. And 20% is eaten by grazers, 80% is decomposed by fire, by fungi, and by bacteria. One really important concept I want you to get, and this is something that I'm going to hit over and over again, is the community physiology. If you look at the physiology of any environment, you will see this pattern. Right? The four most common polymers that you find in the environment are cellulose, chitin, lignin, and protein, and we'll be going through these as we go through this module. These will be depolymerized by various organisms into lipids and fats, sugars and amino acids, and nucleotides. If there's oxygen around, aerobic respirers will dominate, and they will convert all of this into water and CO2. If oxygen is not present, which sometimes happens, syntrophs and other fermenters will convert this into hydrogen and CO2, into acetate then, lactate and other organic acids, and ethanol. This milieu of chemicals, hydrogen, CO2, acetate, other organic acids, etc., will then get acted upon by either methanogens, if sulfate is limiting, to form methane and water, or by sulfate reducers if sulfate is present to be used as a terminal electron acceptor into hydrogen sulfate, water, and CO2. So those are the three degradation pathways. You can either depolymerize stuff, then aerobically respire it, or it will get fermented into acids in a second phase, right? Acids, ethanol acetate, hydrogen, and CO2. This gets acted on by methanogens or sulfate reducers. Now, if you examine this diagram and you think about it, what are the major products under aerobic conditions? Under anaerobic conditions where there's no sulfate present. Under anaerobic conditions with sulfate. Okay, so under aerobic conditions, the major products are water and CO2. Under anaerobic conditions with no sulfate, the major products are methane and water. And under anaerobic conditions with sulfate, the major products are hydrogen sulfide, water, and CO2. All right, so that's the first section in community physiology. And again, remember that. I think it's really important. Hint, it'll be on the exam. What we're going to talk about now is how do we know what's there? If you're a macrobiologist and you're looking at macroorganisms, what you do is you put on your hiking boots, you get your gear on, and you head out into the environment and you start counting the stuff, the living organisms that you see. Well, that's kind of hard to do for microorganisms. If you'd think, well, I'll take a sample, I'll put it under a microscope. I'll look at what's under the microscope. And then the things that kind of look like bacteria are probably bacteria. But wait a minute, well, who's alive and who's dead? Is that thing that I think is a bacteria or is it not? So how do you identify them? One of the ways that this can be overcome is with fluorescent dyes that detect viability. 5-cyano-2,3-ditoyl tetrazolium chloride, which I will call CTC from now on, is a very useful fluorescent compound to use. When it's oxidized, it's colorless. Even when you shine a fluorescent light at it, it won't fluoresce. If you reduce it, it turns red under fluorescent light. It will fluoresce red. And it's also insoluble. So it will float into an organism, and metabolism is a reducing environment. And electrons will actually be added to CTC in this environment from the various compounds that are in a cell. And it will then, first of all, cause it to precipitate, and then secondly, it will fluoresce. So anywhere it fluoresces, you know there's active metabolism going on. So here's an example. This is a biofilm shown here of Pseudomonas putida. First of all, you add a DAPI into it. DAPI is another fluorescent dye. It sticks to and detects DNA, so then when you shine a fluorescent light on it, it turns blue. You then take the same biofilm and you add CTC to it, and this is when it's actively growing, and you can see, if you compare A and B, that all of the cells here are active and growing and they're metabolizing. Now you take and starve that biofilm for an hour so that it shuts off all the metabolism. You stain it with DAPI, the cells are all still there, but now when you throw CTC in there and do the same detection in panel D, you see that, nope, there's no activity there. These cells are metabolically inactive. So you can tell, using CTC, what's metabolically inactive. So now we can investigate the environment. So if you do microscopy using these methods, it turns out most of the things that we thought were cells are actually cells. Those are organisms there. Also, about 1 to 35% of them are actually active. So how do we know this? Well, acrin in orange, again, is a molecule that binds to DNA and detects it, but shows no metabolism. So it's just saying that, okay, there's something there that has DNA. And if you look at any environment, and this is true in most environments, this one just happens to be a soil, you find about 1 billion living things, or things that were living, per gram. However, if you try to culture that, and you wait a few days, looks like 3 or 4 days, and count the colony forming units, it's between 2 to 4 orders of magnitude less organisms there by a viable plate count. Now, if you use these CTC staining methods and you look for active metabolism, you can get this up to about 5 times 10 to the 7th. But that's still 20-fold less than what you're seeing in the Actrogen orange stain. And it's like, what is the difference here? What's going on? What this tells you is our culturing techniques are incredibly inefficient. We're getting between 1.1 and 10% of the organisms in the environment into culture. So most of them are not being cultured. We need other methods to assay the environment and detect what's there. All right, so the techniques that we're going to talk about: fish amplicon sequencing, which is PCR amplification of the 16S ribosomal RNA sequence and then sequencing of it. All right, metagenomics, which is whole sample sequencing, and metabolomics. These methods have revolutionized microbial ecology because it gives it a chance to detect everything that's there, not just the stuff we can culture. So what is fish? Fish is fluorescent in situ hybridization. You design a probe, and this DNA probe will be to a region of interest. And the very nice thing about ribosomal RNA is that there are areas of extremely high sequence conservation that you will find in the whole domain of bacteria. So you can design a probe that goes through the whole domain. There are other areas that are more variable, and some of them that are hypervariable. So you can design probes that will hybridize, right? So this is a piece of DNA that you've made, and you can make this sequence anything you want. You can synthesize any sequence you want of, let's say you want to make a sequence of 25 base pairs or 40 base pairs or whatever. And then you add a fluorescent tag to it. This sequence will then stick to DNA that it can hybridize to, that has a complementary sequence. And again, you can design that probe so it will bind to everything in a domain, like bacteria, archaea, or eukarya. You can design a probe so it sticks to a certain phyla that you're interested in, let's say proteobacteria. Or you can even do it at the species level if you use these really hypervariable regions in the 16S ribosomal RNA. So you take your sample and you stick everything on a slide. You then treat it in such a way that you cause the DNA to denature. Now the DNA is still with the cells on this slide, it's now just denatured. You then throw the probe in, and the probe goes throughout the slide, and it will bind and hybridize to regions that it matches. You then visualize this on a fluorescent microscope. And what you see is anywhere there's DNA that the probe binds to, that matches the group that you're looking at, either like a domain of bacteria or a phyla like proteobacteria or a species. Okay, so with these fish probes, we can go into an environment, look at it, and identify what the things are and what position they are, where they are in the environment. Are they in a biofilm? Who are they with? And that way it really gives us a good idea of who's there and where they are. Another way you can survey the environment and actually get it in much more detail, because the limitation of fish is that you can only look at one section of a slide at a time, is using amplicon sequencing. So in amplicon sequencing, you collect all the cells in the environment, you extract their DNA, you run PCR reactions using primers that flank the 16S ribosomal RNA genes. You then add adapters to this and run it through a DNA sequencing protocol. Right now, a lot of what is used is Lumina sequencing by synthesis, and hopefully you watched the movies from last time. And then you can take these and count. Okay, how many 16S ribosomal RNA sequences do I see of this species, and organize them all and bin them all and then count them all. And you can get a complete survey of the whole population without doing any culturing. So this is very effective. Another thing that you can do is whole genome sequencing. In this case, you skip the whole amplification step, and what you do is you extract all the DNA, you shear it all up, you add adapters, and you sequence it. And you get a huge amount of these small little sequences. It's like having 500, 600 jigsaw puzzles and you've mixed all the pieces together and then you take them apart and you build up those jigsaw puzzles on their own. So from this, you'll take all these sequences, you'll assemble chromosomes, and you'll learn who's there. But not only will you learn who's there and identify them, if you can look at the entire chromosome, you can also start to assemble the genome and start figuring out what the metabolic capabilities of these organisms are. So whole genome sequencing is becoming more and more of a possibility because sequencing is becoming cheaper and cheaper. So probably the way they're going to go as we do more surveys. This gives you an idea of the types of metabolisms that might be going on in an environment because, again, you can start to look at whole genomes, you can start to figure out what various proteins are doing in those genomes, and you can make predictions. Metabolomics actually gives you an insight into what small molecules are there, what metabolites are there, and how they're being converted. And metabolomics is the study of the entire set of small molecules in a biological system. The key to this is using a mass spectrometer. What you do is you collect a sample, you extract all the metabolites, and you inject it into some type of separation apparatus. This could be a gas chromatograph or a LC. What comes off of the LC is then run on a mass spec. So you separate out a lot of the compounds and then you run them on a mass spectrometer. The mass spectrometer can identify what a lot of those peaks are, and from this you can make a metabolic map. If you see all the intermediates of glycolysis in a sample, you know that glycolysis is probably going on in that sample. If you see all the intermediates of tryptophan biosynthesis, you know something's synthesizing tryptophan. From this, you can determine hundreds of compounds in a single run, and again, start to build these metabolic maps. All right, to end this section, let's ask you some questions that will determine how much of this is making sense to you. Okay, number one, you want to determine the microbial community of the Antarctic soil recently exposed by melting glaciers. What would be the best method for determining this? Number two, your team has characterized the community of Antarctic soil and identified a unique microbe. You now want to determine where in the soil structure this unique microbe is found. Which method would be best for this? Question one, since you're trying to ascertain the entire community, the best thing to do in this case would probably be you collect a sample and then do some type of sequencing of it. Amplicon sequencing or metagenomics. That way you would characterize the whole environment. Since this is the first time that you're doing it, using the 16S ribosomal RNA is probably a good idea, and so amplicon sequencing would be the best. If you have an organism that you're now trying to see where it is in the environment, in question two, and you're trying to figure out soil structure, where this thing is placed, etc., then the best method to use in that case would be fish. You'd design a fish probe that's specific for that microbe, you'd add it to your stain slide, and then you'd use a fluorescent microscope to find it. All right, that ends our introduction to microbial ecology. In the next lecture, we'll start looking at various environments.